Today's lesson centers around 6.2, which is chemical reactions. And specifically, when we talk about chemical reaction in living systems, we focus on enzymes, which are your cells' tools to get things done, to allow for and provide um, for chemical reactions. Now, this happens in plants and animals. Anything that's living has these chemical reactions occurring. And specifically, the way that they get done and very effectively is by the use of enzymes. So, if you're looking on page 156 to 160, this is really where the focus is. And it starts off with a chapter looking at a specific chemical reaction that's um, very common in nature, and that is glucose, which is used by your cells. And that has a formula of C6H12O6 plus combination of oxygen gas, O2, and then has a arrow here indicating that this is a chemical reaction. In other words, the molecules on the left here are my glucose and my oxygen are reactants, and the material, in this case my molecules on the right, are carbon dioxide and water, which are the products. So if this is an example of a chemical reaction that occurs within our own tissues, this is called cell respiration, which our cells use glucose or sugars plus oxygen, and it produces the carbon dioxide, the waste products of cells, along with a little bit of water. So those are my products. And it just so happens that in nature, that this chemical reaction is reversible. And in fact, plants use the carbon dioxide produced along with the water to produce more glucose in plants. And that process is photosynthesis, which is another level of this. But if we look at this and how enzymes are used, in other words, these tools to allow these chemical reactions to occur, this is kind of the fun piece. And so as we look at this, we'll focus on enzymes. And enzymes are catalysts. And this particular type of catalysts are made up of proteins. So enzymes are protein catalysts. And a catalyst is, the question is, so what is a catalyst? Well, a catalyst allows for chemical reactions. In other words, it speeds it up. And it does that without changing its chemical properties itself. So, in other words, it can be used over again. Some of these catalysts are made up of organic compounds, and we call those enzymes. So, not all catalysts are enzymes. However, all enzymes are catalysts. So, in a nutshell, we're going to focus on the word enzyme here. And an enzyme is allows for, not is, but allows for these chemical reactions to proceed in each and every one of your cells 24 hours a day, 7 days a week. In other words, this is the tools, this is the hammer, the screwdrivers, the saws. These are the tools of cells to get things done, to construct things, to deconstruct things, to move things around. These all require enzymes. In fact, it's very, very complicated. And the number of enzymes used in every chemical reaction is just mind-boggling. So enzymes do this. They speed up chemical reactions. So if you look below, you'll notice that in red there it says enzymes speed up rates of reactions by at least 1 million times. Now again, I can't think that fast. In other words, it's very, very quick. It's what I would say almost instantaneous. And so if we look at how this works and operates, there's several diagrams, and this is something found very similar to your textbooks. But how an enzyme works is called the induced fit model. And what that means is that we have these enzymes have particular shapes, and they're very specific shapes. And they can bend a little bit. I mean, there's some movement within those to fit some of these molecules. But again, they have to be of a particular shape. So the diagram down below here, you have the enzyme in kind of a grayish black color. Notice the shape of it. It has on the top surface, it has two little shapes that will mold a particular product. And this is referred to as the substrate in green. In other words, not, I said product. This is what's going to happen, what's going to change the cellular structure and the bonds that exist within this substance. The enzyme has another area which is called the active site. In other words, this is where the reactions are going to occur. So if you go from the first part of the model to the middle, where the two are combining, where the green and the gray are combining, you'll notice that my substrate has bonded with it and has a very similar shape to my substrate, my active site on my enzyme. So we call this complex the enzyme substrate complex. It's a temporary scenario which allows the enzymes to kind of break some of the bonds on the upper substrate. And when it does that, it can either break bonds or form bonds. I'll be very specific there. So what we get is a new type of reaction. In this case, I even changed colors to indicate that this triangle and this semicircle have changed so much that we are going to separate those completely. So we get two products at the end, and you'll notice that the enzyme here is stationary. So in my diagram back here, what we would need in order for glucose and oxygen to combine and be utilized, this would be my enzyme. In other words, the arrow indicates the chemical reaction, and this is what's going to be required for an enzyme is going to be needed to transform these reactants into these products. So it's just done a little bit differently on the diagram down below. But that's my chemical reaction demonstrating the same thing. The arrow is indicating the enzyme. So when they do this, we break or make new bonds. And so this is my enzyme right here. We've broken or informed those new bonds to form our new products. And the cool thing about an enzyme is that it can be used over again. I'm going to skip the video here. But so how do enzymes work? Well, what they do, and one of the big selling points of the use of an enzyme is that they lower the activation energy. And that's a key point. So if you look in the diagram here, what you'll notice on the chart is that the energy is on the left and the progression of the reaction, in other words, the time is on the bottom. And what you'll notice is that in in red, we have a chemical reaction. Again, maybe something like I have up back here with glucose and oxygen. And this is the amount of energy is required, whatever level that is. Maybe this is, I'll give it a 10 on the top here. This is the amount of energy required for the reaction to proceed. It's like going to the movie, I suppose. You need, let's say, $10 exactly. $9.99 isn't enough. You need $10 exactly in order to go into the movie. You also need a certain value or a certain amount of energy needed for the reaction to proceed. And then it can proceed into the products as time goes on. Now, what an enzyme does is it lowers this energy. This energy, which we refer to as the activation energy, is lowered. So if you look in green, if we have an enzyme present, the same products, maybe again, my glucose and my oxygen, with the presence of an enzyme, can lower the amount. Instead of maybe the value of 10, maybe it only requires a value of 2. And so therefore it goes immediately and transforms that into my products. That's why it takes place in such a fast period of time. So the big key here is that you'll hear me and others, and along with your textbooks, so enzymes lower the amount of energy or activation energy necessary for this to occur. And that difference is significant so that it proceeds at a much faster rate. So when we look at an enzyme that can't work properly because maybe a change in its shape. And so this can lead to some challenges if the enzyme is somehow the shape of it's changed so that it doesn't produce the products. In other words, here's my enzyme. It has a particular shape. Here's my substrate. But if I heat up and it looks like they're going to fit together, but if I heat up this solution with this enzyme, what happens is that this enzyme gets stretched and no longer works. We call this process denaturization. And if it does work, it can lead to some strange things that can lead to protein synthesis, producing the wrong enzyme shapes. Can also be one of those things, as I said, heat, and even changes in pH. So all these things can change the shape of an enzyme. So we have to be really careful. For example, for those of you that are aware of like a temperature, temperatures are fine to fight off viruses temporarily. But if your temperature, your body temperature rises above a certain degree, let's say 104, 105, that can be potentially dangerous and can only be held for very short periods of time before it does damage to your internal cells, especially your brain and some of the nerve tissue that support the brain and neural functions. But again, mutations, heat, and changes in pH can all potentially change the shapes of the tools or the enzymes. If we look at this particular diagram, I wanted to show how maybe enzymes work best. They work best at normal body temperatures. So our cells have lots of enzymes in those. They work best with a temperature of around 37 degrees or about 98 degrees. So body temperature. A fever causes potentially that to spike to a higher, therefore it'll affect the ability for those enzymes to work, and it will in fact increase their ability to denature, almost like frying an egg, where if it gets too hot, you'll change the egg to a point where you're never going back to its original shape. So that melting, the changing shapes stops its functioning and potentially wouldn't perform the job that was necessary within your cells. So that could be a detrimental thing for sure. Um, the other scenario here is where enzymes can maybe work within the intestinal system. We're doing the rat dissection right now, and if you're looking at the enzymes that are found in the pancreas or even the stomach, we do have some that are present that are the whole job is to break down the food so that you can absorb small enough pieces into the circulatory system to maintain your energy necessary. Well, we know that all these are done by enzymes. And so, for example, we have one enzyme called pepsin. Pepsin is found within the stomach and just outside the stomach in the small intestine as well to some degree. But pepsin really needs a high pH. In other words, I should say a low pH. A pH of 1 to 2 works best for it to work. So as food drops into the stomach, that low pH allows it to be broken down into smaller pieces. If, for example, the stomach's pH rises above, notice what happens as the pH increases. The functionality or the ability for that enzyme to work decreases. It's still working, but maybe at a pH of 4, it's only working at about 25% of its potential. Same thing with um, another one called trypsin. Trypsin also breaks down proteins like pepsin does, but pepsin works in the stomach. Trypsin works in the small intestine, specifically the jejunum. And what you'll notice is that as the raise in the pH going from a 5 to a 6, so almost the optimum pH, it seems like, with pepsin is around 2. Trypsin has an optimal pH of around 7, maybe 7.8, and that's where it's working best. So if that's happening, that means that we can break down more food and particles to be absorbed. So again, if we lower or increase the pH, it will affect these enzymes from operating to their most efficient level. Remember that a pH of 7 is around neutral. Anything above or below that, in this case, would be an acid or a base. So that's really kind of the skimming of how enzymes work and operate. We'll look at water and pH along with some of those other chemical reactions in the next section. Thank you, everybody.